Okay. Hello, everyone, again, and welcome to another online lecture. Our last lecture was covering mitosis, and this lecture will be covering meiosis. This should be your last lecture before your exam. Remember that exam 3, I'm just going to do a reminder now, and then we'll do one in class on Monday. But exam 3 is going to be over chapters. We have 10, we have 11 and 12. I almost got it. We have chapters 11 and 12. We have chapters 7, 8, and 9. So 7, 8, 9, no 10, yes, 11 and 12. Um, so let me go ahead and share my screen and let me get our PowerPoint up. We are going to do this fairly straightforward. I will stop at some point and let you know what your pop quiz will be on, what the question will be for your pop quiz. It'll probably be one question worth 10 points, and I will pick that up on Monday. So make sure that you stay through. I won't be adding videos to this lecture because I don't intend to, because I assume that you guys are going to stop and get snacks and listen again and kind of listen at your own leisure. So I'm going to just kind of get through this straight through, and then I won't have too many, hopefully, interruptions. So let me start here. We're starting on chapter 12 with meiosis and sexual reproduction. So we left off at the end of mitosis, which is when cells need to divide or when we need to have more cells. But we're talking about body cells, autosomes, so cells that are not involved in sexual reproduction. So that can mean our kidneys, livers, lungs, skin, especially skin, right? We talked a lot about how skin has a really high turnover rate. We're constantly touching things, we're constantly moving, we lay in beds, everything has friction. And so our skin always has to have this really high turnover rate. Um, so that is mitosis. Mitosis ends in two cells. Mitosis has two cells at the end, and those cells will be identical to the parent cell. They will be considered clones or identical all the way through. They transfer that entire genome. So the result of mitosis is two identical daughter cells, whereas meiosis, which is a similar process, it is still cell division, but it's going to be a little bit different and have a few more steps. Let me move some stuff around here. Okay, so remember this is about sexual reproduction. So in asexual reproduction, a single individual gives rise to offspring that are identical. That's what we talked about in mitosis. That's what that cloning process is. They take themselves and they make extra copies of self. In sexual reproduction, there is some mixing that goes on. Genetic material gets mixed. I mentioned this last class as well, but that's why you could look a lot like one of your parents, but you won't ever be truly identical to one of your parents. And this happens through something called alleles. You see here at the top this word alleles. Alleles are going to be the things that are that recombination, that genetic difference, the differences that we can share. So when we're talking about somatic, which are body cells, somatic cells of sexually reproducing eukaryotes, like people, like cats, like dogs, like whales, anything that is going to be sexually reproducing. Um, Body cells contain pairs of homologous chromosomes. Now remember, homologous chromosomes are the same chromosomes that are found inside that nucleus, one from mother and one from father. So they're going to have all the same genes on them, but one is going to come from mom and one is going to come from dad. So homologous chromosomes will carry genes of the same characteristics. So when we looked at that karyotype, if you think back to our karyotype lecture where we saw all those little X's lined up from kind of largest to smallest across the screen, um, kind of everywhere on, say we have the first little X and there's going to be two pieces, right? And X has kind of a cross or these two pieces. Eye color will be found at the same point on both of those homologous chromosomes. So say it's 1/3 of the way down on that little X on either side. That's going to be a gene for eye color, and it's found on both parts of a homologous chromosome in the same place. So, homologous chromosomes carry genes of the same characteristics. It could mean that mom is giving blue eyes and dad is giving brown eyes, or mom is giving light brown eyes and dad is giving dark brown eyes. But those genes will be found in the same place on a chromosome. Different forms of the same gene are called alleles. So this term allele is referring to the ability to pass down blue or green eyes, curly or straight hair, crooked teeth or straight teeth, anything. Um, the likelihood to develop type 2 diabetes or not. Any of those things can be found on your chromosomes. You get one copy of the potential to develop diabetes from mom and one copy of the potential to develop diabetes from dad. And those are called alleles because they are not going to be identical. They're just found in the same place on a chromosome. Paired genes on homologous chromosomes may vary in DNA sequences as alleles. So, paired genes, meaning the spot on that chromosome that's for eye color from dad, and the spot on that similar chromosome from mom that's for eye color, may have some different DNA sequences. So, those nucleotides, those A, T, Gs, and Cs, may be a little bit different, right? That's what we would call a mutation. Having a T where there should be a C or a G. Again, as we said, not all mutations are bad, they're just different. So those mutations are going to be the basis of differences in shared traits. That's why we can have so many different unique traits. Traits for height, and it's not just tall and short, but being a little taller, a little shorter, dwarfism, gigantism, all of those can be found in your genes. And those would be different alleles found on those chromosomes in different persons' genes. So those come from mutations and they are the basis of differences of shared traits. So offspring of sexual reproducers inherit new combinations of parental alleles, and that results in a new combination of traits. So your combination of traits that make you exactly who you are completely unique to the ones that made your dad the way your dad is, which are completely unique to the ones that made your grandpa just the way your grandpa is, and so on. Every single generation and every single offspring results in a completely new combination of traits. Even if you have the same exact eye color as your brother or sister, they may still be carrying a slightly different gene on their other chromosome that they could pass down to their children. So just because you see the same color doesn't mean that your genes are reading the same thing. And that's really important because that still means even if you look just like your sibling, and we're not talking about twins today, but if you're looking just like your sibling, that still doesn't mean that you carry all the same traits in an identical pattern on both sets of your chromosomes. So this is what I was talking about in that karyotype. You can see here, these are homologous chromosomes. You can see them standing, there are two homologous chromosomes here. And those corresponding colored patches indicate corresponding DNA sequences. So that little red spot at the top corresponds to the red top on the one on the right. Red top on the left corresponds to red top on the right. Same with the little blue region on the left and right. That means that that's coding for eye color, hair color, height, maybe a disease process, maybe the likelihood to develop a larger nose or vitiligo or anything. I'm not sure what these genes are, but they will match up all the way down on moms and dads. Those genes are found in the same place, even if they don't have the same nucleotides. I hope that makes sense. So sex mixes up the genes of two parents. offspring will always be unique. There will be a unique combination of traits. So, as opposed to our cloning cells in mitosis, we're talking now about having these unique combination of traits, and that's what is something advantageous about sex or sexual reproduction. Diversity offers a better chance of surviving environmental change than clones. We hear about this a lot. I think it's in bananas. We had learned to clone plants. We've learned to clone plants long time before we ever learned to clone um, other complex mammals and stuff. But we were able to kind of clone or inbreed, get to the point of inbreeding bananas until there was no genetic diversity or very little genetic diversity. And so then that meant that if a fungus that attacked those bananas or bacteria found its way into the bananas, there would be no genes in place to make any of the individuals hardy enough to withstand or fight off or not be susceptible to that bacteria or fungus. So if something goes wrong in a banana crop, all of the bananas could die or will die because there's no genetic diversity. There's nothing standing in the way of them being taken over by these other organisms or problems. So diversity offers a better chance of surviving environmental change. Clones are much more susceptible to dying or succumbing to environmental changes. Here are just some images. Obviously, a cute puppy is going to have the mother, and it looks like they've given us the idea here that there's a mother and father dog kind of in the middle, and none of their offspring even exactly look just like them. So there's all that genetic recombination, even though they were all born in the same litter. And then we have our asexual reproduction on this plant here, and it's growing from self, right? It's put out some leaves that then can put out some roots, and it's not actually being fertilized in any different way than through self. So this Red Queen hypothesis, I did not include it in your test, but they're talking about the book by Lewis Carroll, Through the Looking Glass. It says here, the Queen of Hearts tells Alice, it takes all the running you can do to keep in the same place. Kind of to keep up with yourself, right? Organisms must constantly adapt and evolve in an ever-changing environment. This is used to explain the evolution of sexual reproduction. So, through all of the, complex Earth history that we think about, there have been these big catastrophes where so many things might die off, and then that leaves room for other to give rise to new organisms. Um, but they're always out competing. There's only a finite number of resources, and so different organisms might be competing for the same resources. And if they're not able to adapt or evolve to better reach the resources or better access or better utilize the resources, then they're not necessarily going to ensure the survivorship of that species. So they're constantly having to run in place just to keep up with everything next to them, to keep in the same place, just to be able to survive all of these changes, these little changes. We see a kind of classic picture of Darwin discussing finches and beak length. Certain finches could be very closely related, but that beak got a little bit longer, and that meant that they could access food further into a trunk. Or the beak got a little thicker and harder, and so it was able to break through the harder outer surrounding part of a fruit, the skin of a fruit. And those are the changes that are made through sexual reproduction. Those are changes that can help survivorship long term. Sexual reproduction mixes up alleles from two parents. So as we saw, I'm going to go back to that karyotype picture. We'll say mom's chromosome on the left and dad's chromosome on the right. We are able to mix them up a little bit to have that genetic diversity that we're looking for. So, sexual reproduction mixes up alleles from two parents. This involves the fusion of gametes, which we'll get into, which are also known as mature reproductive cells. and then undergoing this process of meiosis. So mitosis was also a nuclear division. Meiosis is a nuclear division mechanism that occurs in reproductive cells of eukaryotes. So eukaryotes, multicellular organisms, and it happens only in the reproductive cells. So in this case, we're talking about sperm and egg. And this is going to be the nuclear division that takes place in those cells specifically. This happens in plants and animals. And so you see some of the things we're used to: ovary, which house the eggs, and testes, which house the sperm. And then here you can see with the plant, there's an ovary, there's a stigma, and then there's the anther, and the anther is where the pollen kind of can fall from. So meiosis will halve the number of chromosomes. So in mitosis, we talked about this, that those cells are going to be diploid, and they're always going to have two copies of the chromosomes. When it comes to meiosis, our final product is going to be something called haploid. You see it here in the middle, Haploid. Gametes, so sex cells, sperm and egg cells, are going to be haploid, and that's represented by just the letter N. In mitosis, diploid is represented by the number 2 with the lowercase n. So gametes are specialized cells that are the basis of sexual reproduction. They derive from something called germ cells. They are all haploid, but they will have other details that are different. And gamete formation differs among plants and animals. Haploid germ cells, so those cells with half of the genetic information, form by meiosis, and gametes form when these cells divide by mitosis. So eggs are female gametes and sperm are male gametes. Similar to mitosis, and this is where I'm hoping you will remember that your pop quiz is going to be to compare and contrast mitosis and meiosis. It's also one of the questions on your exam. Interphase is going to be where DNA replication happens. We still have to make new DNA in order for these sex cells to have access to DNA to pass on to offspring. So there is going to be this DNA replication phase. Then it undergoes something called meiosis 1, which has the same names: prophase, metaphase, anaphase, and telophase, all through meiosis 1. And then it'll undergo meiosis 2, and that's going to include prophase 2, metaphase 2, and so on. So nuclear division that halves the chromosome number in reproductive cells. That's our goal, is we want to have half of the genetic information to pass on to the offspring so the other half can come from a new unique person or individual. This ensures that offspring will have the same number of chromosomes as the parent. So if the egg cell has half of the chromosomes and the sperm cell has half of the chromosomes, you now have a full set of diploid chromosomes in the final organism, or in the final offspring. This occurs in germ cells to produce gametes. Gametes are haploid. Gametes are sex cells, are egg and sperm. So just keep that in mind. This is the term gametes, are sex cells, are sperm and egg. Gametes are haploid, represented by that lowercase n. Their chromosome number is half of diploid. Meiosis is a nuclear division. If you were going to say something that's the same between mitosis and meiosis, they're both the process of nuclear division. DNA replication occurs prior to meiosis. That would make sense. We still want offspring to have DNA, and we still need to make sure our cells hold on to DNA. So we need to do some replication. DNA replication occurs prior to meiosis. The nucleus is diploid with 2 sets of chromosomes, one from each parent. During meiosis, chromosomes of a diploid nucleus become distributed into 4 haploid nuclei, and we should see it in an image coming up. Meiosis halves the diploid chromosome number to the haploid chromosome number for the gametes. So in this first step, we have duplicated our DNA, we have replicated our DNA. Then those separate into chromosomes that are diploid in level 2 here, and then those will separate again into 4 haploid cells. So fertilization restores the chromosome number. So once the two sex cells interact to create a new offspring, in this case, a zygote is the early stage of that offspring, then it becomes diploid again. So haploid gametes formed by meiosis. We're duplicating our DNA, replicating our DNA, and then we're separating it and we're separating it again. So we have half of the number of chromosomes needed for a full individual. When two haploid gametes fuse, a zygote is formed, and the first cell becomes of a new individual is formed when sperm and egg meet. Let me see here. I am gonna just pause for 1 second. Okay, let's continue on. Fertilization restores the chromosome number. If meiosis did not precede fertilization, the chromosomes would double with every generation, which we don't want. Yes, we talked a little bit about the benefits of some excess DNA, but not 4 times and then 8 times and then 64 times because it would double every single time, right? If meiosis did not precede fertilization, the chromosome number would double with every generation. So 32 and so on and so on. Chromosome number changes can have drastic consequences in animals. So it is very important that each individual organism, each organism or species has the exact right number of chromosomes. It doesn't mean that every single organism has the same number. I think we looked it up last time and we saw that butterflies have like more chromosomes and tigers have less or something like that. But we looked it up and even other mammals could have different number of chromosomes than us. Chromosome number changes can have drastic consequences in animals. An individual set of chromosomes is like a fine-tuned blueprint that has to be followed in order to have normal functions. And here are just some images of that process of making a zygote. So meiosis 1, prophase 1. So this is the first, we've already done our DNA replication. We've had our checkpoints. We've done all that. We're getting ready to move into meiosis 1, which is going to be that first division. Homologous chromosomes will condense, pair up, and swap segments. Spindle microtubules attach to chromosomes as the nuclear envelope breaks up. So the same thing you could say during prophase 1 in both mitosis and meiosis. The nuclear envelope breaks down. Keep in mind, homologous chromosomes condense. So remember, they're not noodles anymore. They become that nice round kind of tunnel that we looked at, that cylinder. And they will pair up and then swap segments. The homologous chromosome in metaphase 1, homologous chromosome pairs are aligned midway between spindles. And anaphase 1, the homologous chromosomes separate and head towards spindle poles. Telophase, the two clusters of chromosomes reach the spindle poles and a new nuclear envelope forms around them. So 2 haploid nuclei form. In meiosis 2, the chromosomes condense again. The nuclear envelope breaks up. Spindle tubules attach to something called sister chromatids. And we'll see what those look like here in a minute. Metaphase 2, The still duplicated chromosomes are aligned midway between the poles of the spindle. All sister chromatids separate, and now they head to the spindle poles. A complete set of chromosomes cluster at each spindle pole. A new nuclear envelope encloses each cluster, and we have 4 haploid nuclei form. So that was the quick run through, but this is what we're actually talking about. So keep in mind, we're gonna say we have the pink or red ones are chromosomes from mom and the blue ones are chromosomes from dad. In prophase 1, homologous chromosomes condense, so they've become this kind of tubular shape. They pair up. So we have 2 X's, because remember, these were diploid cells. They started as diploid cells. So they will pair up, swap segments, spindle microtubules will attach, and that way they get ready to move and separate. During metaphase 1, homologous chromosomes, and when we're saying homologous chromosomes, we are talking about these kind of full set of chromosomes that you see here, this doubled full set of chromosomes. They're aligned midway between, and the tubules attach 2 chromosomes of each pair. Anaphase pulls them apart, and they are now being separated, but we have 2 diploid cells at the end of telophase. I'm sorry, 2 haploid cells or nuclei at the end of telophase 1. I wanted to go back to prophase. I'm not sure why they didn't include that here. But during prophase 1, when they say swapping segments, that is a term called crossing over as well. And that is truly the eye-colored genes found their way near each other and they switched from one chromosome, from mom's and dad's chromosomes. They switch places. And so you can see now on this kind of larger chromosome in prophase 1 that the little tip of it has traded places. And now that will find its way into new daughter nuclei. So at the end of telophase 1, a complete set of chromosomes clusters at both ends of the cell and loosen up. A nuclear envelope forms around each set to produce 2 haploid nuclei. Then during prophase 2, the chromosomes condense again, spindles form again, each sister chromatid breaks up. The still duplicated chromosomes are aligned, and eventually those are pulled apart. So we have at prophase 1, we have a diploid cell, and now we've made extra copies. And so we have our diploid cell that we've started with, sorry, at the beginning at prophase 1. At the end of telophase 1, we have 2 haploid nuclei. Prophase 2 starts, and we want to break those down even further so that way we don't have a complete genome. We don't want one single genome. Like we don't want a full one. We want a half of a genome. So here you see prophase 2, the chromosomes condense, spindle microtubules attach. In metaphase 2, the still duplicated chromosomes are aligned, just like everything else we've seen with metaphase. And during anaphase, they pull apart. Our sister chromatids pull apart. So, and then at the very end of that, we have telophase 2, and a complete set of chromosomes clusters at both ends of the cell, and a nuclear envelope forms around it. So now we have 4. If you look here, and I want you to just kind of walk through this image set with me. We have our diploid nucleus here in prophase 1. We align them, we pull them apart, and we have 2 at the end, 2 haploid, because we don't have all of mom and dad's genes now in each nuclei. Then we move on to the next nuclear division. And we had some crossing over happen in prophase 1. So now we have a new mixture of genes finding their way into these separate areas. If you look here at anaphase 1, we call them sister chromatids because we're pulling apart the single genome that was left. We're pulling that apart again. So we first pulled apart mom's genes and dad's genes after they shared a little bit, and now we're pulling apart a genome by itself. And that ends up in 4 daughter cells. And if you take a look at these 4 daughter cells, they are not identical. We have a big long gene from dad and a short gene from mom. We have a big long gene from dad with a little bit taken from mom and a short gene from mom, and so on. So those last 4 will not be identical. Once crossing over happens, which is in during prophase 1, crossing over happens, the swapping of DNA segments, the exchange of DNA information across mom and dad's genes to make a totally unique set of cells happens during prophase 1, and we can see that that leads to 4 unique daughter cells at the end of telophase 2. If all the rest of that didn't make sense, I think that was the best explanation for that. So 2 events in meiosis introduce novel combinations of alleles, new combinations of alleles into the gametes. That crossing over in prophase 1 that I just mentioned, and the segregation of chromosomes into gametes. So if there was a question that was like, what are the 2 events that introduce new or unique combinations of alleles? You would say crossing over and the segregation of chromosomes. Along with fertilization, these events contribute to the variation in combinations of traits among the offspring of sexually reproducing species. So then they get fertilized with someone else so the egg has now been fertilized by someone else who has a totally unique set of DNA in their sperm cells, and that gives rise to a completely unique organism, a completely unique individual. This contributes to variation. So variation is what we want. Diversity is what we're looking for, because that helps to kind of keep the genes different and give us a better chance of survival through evolution. So crossing over, which we talked about a couple times, is in prophase 1. Chromatids of homologous chromosomes condense and align along their length and exchange segments. I'm going to include some videos on what crossing over is. I'll make sure to link them at the end. I'm going to include some stuff on crossing over so you can see what that looks like. But this is a process where that karyotype, those little X's, line up next to each other, and then they trade little pieces of their DNA. But the eye color changes with the eye color. The likelihood of freckles changes with the likelihood or non-likelihood of freckles. So chromatids of homologous chromosomes condense and align and exchange segments. This actually exchanges between the 2. This is what we were talking about in that process of meiosis. We have dad's genes and we have mom's genes. Now, something I want you to look at in this image is how the alleles are represented by the letters A and B. Capital A and lowercase a are representing a gene for something. We'll say a gene for being 6 feet tall. And little a, or for being tall or short, we'll go with that. And we'll say that letter B is for eye color, just to keep it simple. So you see that these can be found at the same spot on mom and dad's chromosomes and on the sister chromatids as well. So we have the homologous chromosomes, that's the blue one versus the red one, and we have the sister chromatids. Those are the 2 halves, 1 half each of the X. They then align. You can see here the little tip at the base of letter B crosses over, and then on a different one, it crosses over a little higher. Now, at letter C, we've traded a capitalized B for a lowercase b on the homologous chromosomes. When homologous chromosomes separate in meiosis 1, one of each chromosome pair goes to each of the 2 new nuclei. For each chromosome pair, the maternal or paternal version is equally likely to end up in either nucleus. So it's also random chance which one will end up fertilized. I know I don't know which genes are happening at any given time through any cycle, and I'm sure none of you do either. So we're not picking which genes for eye color or hair texture will make it into that offspring. So it is random. Human gametes have 23 pairs of homologous chromosomes. I'm going to say it again. Human gametes have 23 pairs of homologous chromosomes. Each time a human germ cell undergoes meiosis, the four gametes that form end up with one of, we see our number here, possible combinations of homologous chromosomes. That's a big number. That's a lot of possibilities. And crossing over further increases the genetic diversity. The chance that the maternal or paternal version of any chromosome will end up in a particular nucleus is 50%, because that is the chance that any, because there was a 50%, it's fine. 50%, just trust the number. I won't get too far into it. The spindle randomly segregates the homologous chromosome during meiosis 1. In prophase 1, chromosomes are attached to spindle poles, and each homologous partner becomes attached to the opposite spindle pole. So you can see that here. This is just some different ways that the chromosomes can move through that process and how they can separate or segregate in this case. Mitosis and meiosis. There are obviously we've some parallel stages or parallels between the different stages that we've talked about, prophase, metaphase, anaphase, and telophase. Many more similarities exist at the molecular level. So meiosis may have evolved by remodeling the existing mechanisms of mitosis for repairing damaged DNA. So we talked a lot about how we have these different ways that our body can check to make sure that only healthy, complete, perfect cells move on into the metabolism, into the metabolic process. And meiosis might have been, is being posited as a possibility of being one of those checkpoints at one time, and allowing for some of this diversity ended up being an advantage. And so then that process took off and was able to produce lots more organisms. Here is a nice little comparing chart for you all to look at. Evidence for this hypothesis includes a host of shared molecules. In this case, they're talking again about those breast cancer genes. By fixing the problems with DNA, these molecules maintain integrity of the cell's chromosomes. Keep in mind, I'm not going to ask you too much about this specifically in the test, but it's mostly just saying that the products of the BRCA genes made by all eukaryotes. So fixing the problems with DNA or those mismatched base pairs, um, instead of having like, you remember, like a single switched base pair probably isn't going to lead to some horrible problem, but it could. And BRCA genes are, I believe, a base pair switch. Um, but in this case, the BRC, so we're, sorry, I just heard the baby cry. So by fixing the problems with DNA, these molecules maintain integrity of the cell's chromosomes, meaning that we're not lengthening and lengthening and are having these frame shift mutations. And meiosis allowed us to pare down the number of genes and make sure that we keep that kind of perfect number of genes, through potentially these kind of checkpoints and other things. But this is all much more complicated. I'll send a video out, but that's a much more complicated process than they can explain in this one. So that is it actually for this. We got done with it quicker than I expected. Um, feel free to look through. Let me know if you guys don't have access to the PowerPoint. Let me see here. All right. Perfect timing because the baby started crying. So your quiz will be compare and contrast. So I want you to list two to three similarities and two to three differences, or at least the most important differences between mitosis and meiosis on your upcoming pop quiz and on your upcoming exam. That will be one of your two short answer questions. So make sure that you prepare those answers, and I will see you Monday.